Hello, greetings Imagion shareholders and stakeholders. I'm Isaac Bright, Managing Director and CEO here at Imagion Biosystems, and I'm happy to share with you the data and the presentation that I presented earlier this week at the San Antonio Breast Cancer Symposium. This meeting is largely attended by world-leading and thought-leading clinicians focused on all things breast cancer. A lot of industry participants are present, but it's oriented very much towards how do we advance the state of the art and the state of medicine in addressing unmet needs in the breast cancer market in particular. And so there are a number of very technical aspects of how a lot of the presentations go, including and especially in clinically advanced programs, such as the success we've had with MagSense HER2 Imaging Agent. This particular poster presentation is not part of the main event, if you will, at San Antonio Breast Cancer Symposium. Again, we have accomplished a remarkable feat in establishing safety and clear feasibility demonstration of the clinical utility of molecular MRI using MagSense HER2 Imaging Agent. But this was a poster presentation, so I was allotted a total of 4 minutes to deliver this presentation, and obviously had great opportunity to have exchange around the poster after the presentation material had been delivered. And we continue to have important dialogues with participants throughout the value chain in diagnostic imaging and precision oncology, focused on our opportunities to improve the standard of care with MagSense HER2 Imaging Agent. That said, as we only had 4 minutes to do this presentation, it is scripted, and I will deliver the presentation according to the script that I used when I presented these data on Wednesday. So without further ado, let me please review for you the full final results of the MagSense HER2 Imaging Agent clinical trial presentation at the San Antonio Breast Cancer Symposium earlier this week. Importantly, at most scientific sessions, there's always an important opportunity for presenters to make financial disclosures. Conflicts of interest may arise, and obviously I am compliant. I wanted to let everyone know at San Antonio Breast Cancer Symposium I'm a proud employee of Imagion Biosystems and excited about the opportunities that we'll present for the company. Some of the discussion in this material is forward-looking, and as a listed company on the Australian Stock Exchange, I need to disclose where we are with regard to public disclosures, forward-looking statements that I'll make, and the fact that investing involves risk. MagSense HER2 Imaging Agent is a superparamagnetic iron oxide nanoparticle biofunctionalized with trastuzumab to target HER2-positive breast cancer cells. The MagSense HER2 Imaging Agent is indicated as an adjunct to MRI for the assessment of axillary nodal disease in patients diagnosed with HER2-positive primary breast cancer. This first-in-human study of MSH2IA has been completed at 4 clinical sites in Australia, evaluating the safety and preliminary feasibility to detect metastatic disease in HER2-positive breast cancer patients. Participants had a likelihood of lymph node metastasis in the judgment of the investigators. All participants received a single dose of the drug as a subareolar or peritumoral injection. Baseline MRI assessments were performed within 3 days before drug administration, and a second MRI was performed at an additional time point 18 to 78 hours after MSH2IA administration and before any neoadjuvant therapy. The primary objective was to demonstrate the safety and tolerability of the drug. As an exploratory objective, the study evaluated the potential for MSH2IA molecular MRI to assess if the lymph nodes draining the site of a HER2-positive breast cancer are HER2-positive or HER2-negative. A total of 13 participants were treated under this protocol. These patients were all female and primarily white and not Hispanic or Latino, with a mean age of 57 years. Participants primarily had unilateral invasive ductal carcinoma, clinical stage 2A or 2B. The mean time since diagnosis was 21 days, and the mean tumor size was 32.4 millimeters. The majority of cancers were HER2 positive, ER positive, and PR negative. Safety assessments included incidence and severity of AEs, incidence of SAEs, clinically significant changes from baseline in laboratory parameters, and injection site reactions. MagSense HER2 Imaging Agent causes changes in the relaxation properties of the normal tissue being imaged, resulting in image contrast on MRI. For normal lymph nodes, the drug is taken up by resident macrophages, resulting in relatively uniform hypointense contrast. When tumor cells have metastasized to a node, because MagSense HER2 Imaging Agent is a molecularly targeted nanoparticle, specific binding between the target on the mets and the ligand on the drug results in heterogeneous hypointensity, where the nanoparticles have become bound to the tumor cells in the invaded nodes. The Central Imaging Lab, with 2 independent reviewers, performed blinded review of the MR scans. Nodes were assessed by both conventional radiological measures, such as size and morphology, as well as for changes in contrast intensity between pre-dose and post-dose MRI scans, and the discriminating factors such as homogeneous versus heterogeneous hypointensity patterns. A key component of the study was the collection and analysis of the dissected or biopsied lymph node tissue. Specific staining and histopathology were conducted by the Central Pathology Laboratory, including H&E staining, Prussian blue staining, and HER2 immunohistochemistry as per College of American Pathology guidelines. MRI-based assessment of pre-dose versus post-dose MSH2IA injection was possible in 8 of the 13 patients. There were image artifact issues or agent uptake issues in the remaining 5 patients. Radiologists' assessments indicate uptake of the drug by both normal and tumor-containing nodes. In normal nodes, the uptake of the agent results in a uniform dark contrast hypointensity through the entire node. In tumor-containing nodes, the radiologists identified scattered regions of speckled appearance indicative of a heterogeneous architecture with partial irregular darkening of the nodes. MRI assessment of post-dose MSH2IA imaging performed similar to or better than the standard of care axillary ultrasound imaging in all 8 of these participants. Post-dose images provided better clarity in resolving indeterminate nodes, falsely called normal, and suspicious nodes compared to conventional MR imaging. And 7 of the 8 patients with suspicious nodes by post-dose imaging were also positive by pathology, demonstrating patient-level concordance. These data qualitatively suggest that MSH2IA aids in the identification of axillary lymph nodes and aids in the discrimination of tumor-containing lymph nodes from normal lymph nodes without safety concerns for participants. Of course, we'd like to thank our investigators in Australia and our patients for their contributions to this work, and we're thrilled with the progress we've made advancing and completing our phase 1 study of MagSense HER2 Imaging Agent. I am so thrilled to be building Imagion Biosystems with the team of Imagineers here in San Diego and in Australia, and we look forward to continued success developing this drug and the rest of our pipeline for MagSense molecular MRI to become more of reality and drive the best possible clinical impact we can achieve. Thank you.